Okay, so sorry for the delays. I've been trying to get this up for a little while. There are some kinks that I've had to work out. I think I'm through them now. So my plan is that in the future you will receive all new information on Monday. I just couldn't meet that this week, so I apologize. So when we left, we were talking about replication. And so now we're going to shift gears, move away from replication. So that is using DNA in order to make new copies. We're gonna move away from replication and think about transcription. And so there, we're considering how we use DNA as a template in order to make RNA. So I hope you appreciate at this point in the semester that I like to start kind of big picture. I want to tell, sort of frame things, help to get you to think about where we're going over the next couple weeks. And so in order to do that, I've put together this next slide. So we're going to talk about transcription. We're going to talk about transcription in 5 parts. So this video is going to cover the basics of transcription. So we'll talk about the transcription bubble. We'll talk about template strand versus non-template strand. Some other kind of big picture details about transcription. So these are things that apply to transcription in any type of cell. Before the week's over, or as part of this page, we'll talk about transcription in bacteria. So there are some specifics about transcription initiation and elongation in bacteria. There are also some specifics about termination in bacteria that we'll cover. And then next week, we'll come back and talk about transcription in eukaryotes, and we'll talk about RNA processing. And so there will be a little bit of RNA processing that we don't cover next week. And so then the third week will be wrapping up RNA processing in eukaryotes, and then talking about some RNA specific techniques. So hopefully all of that makes sense. We're going to move through all of these 5 parts, and then by the end, it'll be time for exam 3. So we're thinking about transcription, and so we're thinking about RNA. And so here, I'm just kind of refreshing your memory. So we're talking about ribonucleotides. So these ribonucleotides have a hydroxyl on the 2' carbon. If we were in class, I would poll everybody and you would all announce to me, 2' carbon, right? That's how I hear it in my head. There's a hydroxyl group on the 2' carbon. So the 1' carbon is here, the 2' carbon is here, the 3' carbon is here, 4 is here, 5' carbon is here. And so again, we've got the phosphate group attached to the 5' carbon, that nitrogenous base attached to the 1' carbon. And so this is no different from deoxyribonucleotides. What is different is that we've added a hydroxyl group on the 2' carbon. So this is not a nucleoside. Nucleosides are composed of just the nitrogenous base and the sugar. This, what we're showing, is a nucleotide because it has a phosphate group attached. And so this is adenosine 5' monophosphate. We've got 1 phosphate group here, 5' monophosphate. We can abbreviate that as AMP or A. Okay, so adenosine, we're not talking about the deoxy form, we're talking about the ribonucleotide, adenosine monophosphate. So we could go down the line. There's guanosine here, cytidine here. Uridine is very similar to thymidine that we saw in DNA, or deoxythymidine that we saw in DNA. The difference is that uracil, the nitrogenous base up here, has lost a methyl group that's attached to the thymidine. So uracil is very similar to thymine. So uridine is very similar to thymidine. And you can see down here that we've got adenine bonding to uracil. And so we would expect Watson-Crick pairing to apply to RNA, except instead of bonding A to T, we're bonding A to U. So G bonds to C with 3 hydrogen bonds, and A bonds to U with 2 hydrogen bonds. The last point I want to make before we walk away from ribonucleotides is that RNTPs are 100 to 1,000 times more abundant than DNTPs in a cell. And so in the cytosol and in the nucleus, we would expect to see a lot more of these kinds of nucleotides than deoxyribonucleotides. Okay, so again, all of that should be review. Just trying to get the juices flowing again. It's been a couple weeks off. So if we're thinking about RNA, there are 3 kinds of RNA that we could talk about. And this applies to, again, both bacteria and eukaryotes. So there's rRNA, or ribosomal RNA. So in yellow, we've got ribosomal RNA. And we've got messenger RNA, or mRNA, down here in green. And then this dark green color right here is transfer RNA, or tRNA. And so what I'm showing you here is the process of translation. So we've got transfer RNA packaged and bound to messenger RNA. And then we've got ribosomal RNA that's sandwiching either side of that messenger RNA. So all different types of RNA present here, kind of sandwiched in between the rRNA. So replication is not selective. And so what that means is replication is going through, it's copying everything that's present in the genome. Transcription, by contrast, is selective. And so there are only particular regions of the genome that will be copied as part of transcription. And so the RNA down here is being made 5' to 3'. So we're adding a new RNTP on this end. So the 3' hydroxyl here is presented, the new RNTP comes in and binds. The RNA, importantly, is hydrogen bonding to a strand that we call the template strand. And so the template strand would be complementary to the RNA that's being made. The non-template strand, so this whole thing right here is the transcription bubble. So the transcription bubble is what's being separated, or where the DNA is being separated. We've got RNA that's part of the transcription bubble. On the other side of the template strand, so the non-template strand up here, we call this the coding strand, or the non-template strand. You can think of those things as being interchangeable. So transcription bubble here, non-template strand up top, this is moving 5' to 3'. If the direction of transcription is from left to right, we're going 5' to 3' like this. The template strand is 3' to 5' like this. And the RNA is 5' to 3' like this. Make sure you can picture the orientation of each of those strands. And so make sure that you can understand that non-template or coding strand is going to be complementary to template strand, and that RNA is going to be complementary to the template strand as well. Okay. So there are regulatory sequences that mark the beginning and end of transcription. So we've got promoter here and a terminator here. We're always going to move from promoter to terminator. So 5' to 3', this is our RNA up here. And what we're marking, the promoter to terminator here, is marking the template strand. And so we're thinking about right here, this green area is going to be the start of transcription. This purple area will be the stop. So 3' to 5', we're moving right to left. That's showing you where the template strand is. Likewise, down here, we've got a different template strand pointed in an opposite direction, so we're moving left to right. And so this is, the point of this picture is twofold. One, we're always going to move from promoter to terminator, and two, the promoter and terminator could be opposite directions, right? And so each gene is going to have a different direction on the chromosome. So here we're going from right to left, here we're going from left to right. And so the gene that's present here is always going to be transcribed the same way, but this gene doesn't have to be transcribed the same way as this gene, right? So if this is gene A, it doesn't have to work the same as gene B. So on the last slide, I talked to you about the complementarity. So we've got coding, template, and RNA here. I'm just showing you on the level of the nitrogenous base. So ATG, the template would be TAC, and the RNA would be AUG. So the really convenient thing about the coding strand or the non-template strand is that we can take the sequence as it's presented, 5' to 3', and we can swap out the T's for U's. And so in a laboratory setting, we think a lot about the coding strand, even though it's not actually involved in the process of transcription. And the reason we think about the coding strand is because it's already got the complement present in it, right? So we don't have to think about anti-parallel. We don't have to think about Watson-Crick base pairing. We just take the information that's presented to us, 5' to 3', and we swap the T's for U's. And so by convention, the promoter, the gene, and the terminator are going to be documented. So if you go into NCBI and look for this information, which is what you've done as part of the sequence analysis activity in lab, if you go into NCBI, you'll see that the promoter, terminator, gene, they're all being presented to you as the coding strand, even though we understand as a group that that's not actually the strand that's involved in making the RNA. So we thought at this point about the little pieces, right? We thought about the RNTPs. We thought about the larger pieces, the nucleic acids. And so the next thing we need to think about is the polymerases that are involved in making this happen. And so I want to start with some similarities between DNA and RNA polymerases. So DNA polymerase and RNA polymerase are both multi-subunit proteins. So we didn't focus on that too much when we talked about replication, but there are multiple subunits that come together in order to move replication forward. Here we've got multiple subunits, so each of these has a separate end, right? These are separately translated proteins that are all coming together. So this is a quaternary structure, if you remember from the first unit. We've got multiple subunits that are sticking together, bound to one another, and they function as a unit altogether. So two more similarities. Both are catalyzing addition of nucleosides triphosphate using a free 3' hydroxyl. So at this point, you should just appreciate that as dogma, right? A free 3' hydroxyl is where the new nucleosides triphosphate will always be added, whether we're thinking about RNA or DNA. We're also using magnesium to catalyze the reaction. So there's a divalent cation that's involved in catalyzing the addition of a new nucleoside triphosphate. The last point I want to make with this slide is that the structure, so I've got ribbon diagrams here showing you bacterial RNA polymerase and eukaryotic RNA polymerase. The structures are very similar to one another. And so you could overlay this with this and you would see similar, yeah, they have a lot of similarities. So one thing worth pointing out here is that we've got one kind of bacterial RNA polymerase. There are multiple kinds of eukaryotic RNA polymerases, but I want to kind of de-emphasize that now. We'll get back into that when we talk about eukaryotes next week. So the transcription bubble looks like this. We've got nucleic acids here. We're putting our RNA polymerase here. What you may have noticed on the last slide is that the RNA polymerase looks kind of like a claw. And so we've got the pinchers on this end here. The pinchers are facing in the direction of transcription. And so the small end of the pinchers is toward the DNA that is to be transcribed. So here we're moving left to right, pinchers in the direction of transcription. There's a transcription bubble here. And so the transcription bubble is made by a little pin that's present in the RNA polymerase. It's ripping through and separating those strands and making that transcription bubble. In bacteria, it's about 17 base pairs of unwound DNA. And as you would imagine, unwinding the DNA induces positive supercoiling on one end and then negative supercoiling on the other end. So if the direction of transcription is from left to right, we would call this downstream DNA. So the direction that the RNA polymerase is moving is downstream. And we would call this upstream DNA. And so The polymerase is moving downstream and it's inducing positive supercoiling downstream. And then upstream, it would be inducing negative right-handed supercoils. So if we're thinking about where the RNA is actually physically binding, there are going to be about 8 base pairs of complementary DNA and RNA, or RNA-DNA hybrid. There'll be going to be about 8 base pairs of contact between the RNA and the DNA. And then the last number to kind of throw at you as part of all of this is that the polymerase is moving at a rate of about 50 to 90 nucleotides per second, so pretty fast. Okay, the last slide before we wrap up for this little video, before we take a break and walk around. The last slide is showing you that we've got new RNTPs coming in a way that's very similar to what we saw with replication. So when there's a match between the template and the incoming RNTP, we get close contact between this nucleophile and the alpha phosphate. The magnesium is helping to hold this as a negative oxygen. It's when the hydrogen pops off, it's encouraging it to stay as a negative oxygen. It's encouraging it to keep its extra electron. This makes it a strong nucleophile, which means it can reach out and grab this phosphate. Just like what we saw with DNA, this magnesium is helping to stabilize the leaving group. And so the combination of these two magnesiums is encouraging this reaction to move forward. So we've got a new RNTP that's coming in. The 5' phosphate group is involved in is being attacked by the 3' oxygen, and we're forming a new bond as part of that. So one thing that's different between RNA polymerase and DNA polymerase is that with RNA polymerase, we don't need a primer to start the reaction. So 2 RNTPs can come together de novo. There is no primer required. And the last thing is that RNA polymerases lack a separate proofreading site. And so errors are more likely. But I built a question in that will help you think about why that's tolerated. So I hope all of this makes sense. Again, please reach out to me if you have questions. There's a lot more content to go through, but I hope you stand up, take a break, do something else, and come back and finish the other videos. Thanks, y'all. See you soon.